Assalamualaikum and hi guys. Today your topic is about plant hormone. So under plant hormone we have five different types. Under five different types they have many or huge of the information. For the first plant hormone you will learn about the cytokinins. Alright, so we look on their learning objective. So for the learning objective, cytokinin, you need to know about the synthesis of the cytokinin. They actually from the compound that we call as adenine, and where actually the location in the plant that synthesis this kind of plant hormone. Second learning objective is you need to know what types of the mechanism that able to control the bioactivity of the cytokinin. The next one, you need to know also how the cytokinin are transported within the plant after they are synthesized in the root tips or in the immature seed. The next one is you need to know what the roles of the cytokinin or what is the function of the cytokinin. And we are more focused on their respect to the cell division and the senescence. And the last one, the LO is regarding to you need to recognize the receptor for the cytokinin and the signaling pathway activated by cytokinins. For their backgrounds, you need to know that the cytokinin are derivative of adenosine monophosphate. As I mentioned before in the first LO, this cytokinin is coming from the adenine. Okay, means they actually is coming from the adenosine monophosphate that we call also as AMP. If we talk about the cell division, okay, or the energy mechanism, okay, you may found this kind of AMP. So under the cytokinin, if we look on their structure, we can divide this plant hormone as two names, which is if their side chain is related to the isoprene, so we call that cytokinin as isoprenoid cytokinin. If their side chain contain an aromatic ring, okay, not the isoprene, we call this cytokinin as aromatic cytokinin. So you need able to identify when you look on the structure of the cytokinin, you able to distinguish is it this is isoprenoid or aromatic cytokinin. For the plant hormone, they have the natural plant hormone or the synthetic plant hormone. Okay, so for the natural plant hormone, for the isoprenoid cytokinin, okay, we call or example is N6-isopentyladenine or IP transitin TZ or dihydroxythin TZ. This is the structure of the cytokinin as the natural aromatic cytokinin. Okay, because we have two, we have isoprene cytokinin and aromatic. So this slide showing you about the aromatic cytokinin, okay, with their structure. So the first, not the first, actually one of the natural aromatic cytokinin, example benzyl adenine, or we call also as BAP. However, the first cytokinin that have been discovered in our research was the synthetic that derivative from the kinetic. That kinetic is actually the natural. However, the first cytokinin that discovered is actually the synthetic that derived from the kinetic hormone. However, from the previous study, this kinetin is not actually detected in plant. However, that is reported in the human urine. And if you look on the illustration here, you can see a different name of the cytokinin hormone, which is, we can see we have the kinetin, benzyladenine, tetrahydropyrenyl, and diphenylurea. So all of this is under aromatic cytokinin. Okay, so this one is not only for the aromatic cytokinin but also isoprenoid cytokinin. Okay, so if you look on the structure for the kinetin and BAP, they do not have the isoprenoid in their side chain, but they have the aromatic group. So because of that, these two hormones, kinetin and BAP, we can categorize them as the aromatic cytokinin. However, for the IP and also the Z-transitin, they do not have aromatic group, but they have the isoprenoid in their side chain. So we group them as the isoprenoid cytokinin. And here is all the combination for the aromatic and also the isoprenoid cytokinin under the natural cytokinin. So they have a lot of the compound under the natural cytokinin. Alright, so now we look on how the cytokinin produce, or how the cytokinin synthesis. So if we look on the pathway, okay, what we can say that the synthesis of the cytokinin, they begin with the compound that we call as AMP, as I have mentioned before. Okay, for the cytokinin, they actually derivative from the AMP. So this process, if you look, they have a complex structure, but actually they have a simple step. Okay, where you can see this process we call as two subsequent steps because they have two steps. First step is to remove the phosphate group, and second step is to remove the ribose. Okay, so please look clearly. Every step is different. First is to remove phosphate. Second one is to remove the ribose sugar. So you can see AMP remove phosphate, remove the ribose, they produce the AIP. They produce the IP, which is N-isopentyladenine. But they have another reaction, another pathway, which is from the AMP also they produce the zetin, which is different cytokinin hormone. So in this process, okay, the isopentenyl side chain can be hydroxylated, okay, along with the removal of the phosphate and the ribose to form a zetin. What is the hydroxylated? If you look on the production of IP compared with the zetin, okay, for the production of the zetin, they need to enter, or they need the oxygen. So because of that, this process we call as hydroxylated. Okay, so second process, first they remove the phosphate, second process they remove the phosphate and ribose, and at the same time they are hydroxylated, okay, entering of the oxygen in order to produce the zetin. So what we can see that cytokinin in this process can be converted from the free base form to a riboside or ribopine because they remove the ribose sugar. Okay, now we look on the third LO, or third learning objective for this topic. So that is regarding to the mechanism that you need to know in order you able to control the cytokinin activity, or in the simple words, you able to control the function of the cytokinin. So the first process we call as conjugation. What means by conjugation? Conjugate means you join. Okay, join between the cytokinin with the glucose or cytokinin with the amino acid. So this slide explain to you regarding to the conjugation with the glucose, which is when they are joined with the glucose, they will produce the O-glycoside of cytokinin, okay, that is inactive, and they serve as the storage form because they're not active, so they serve only as a storage. So when they need back the cytokinin, okay, for example, transitin conjugate with the glucose become the O-glucosyl transitin, and this O-glycosyl transitin we call as inactive transitin, and it will become a storage form of cytokinin. However, when the condition need back the transitin, so this O-glucosyl transitin able to hydrolyze by the enzyme that we call as glucosidase. So they able to restoring back the activity of the transitin. Alright, so means in the overall process, this conjugation with the glucose is not permanently inactive, just only temporary inactive form of the cytokinin. So if you are confusing in this illustration, transitin able to oscillate become the cis-cytin. Of course, this is a form 2 form of the zetin. It can be as a trans or as a cis. However, a similar process of the conjugation. They should be conjugate with the glucose and it will be formed inactive of the zetin, either for the trans and for the cis. However, okay, the conjugation not only occur only with the glucose, but they also occur with the nitrogen. Okay, atom in the purine ring, so they will be formed N glucoside. So this explanation in the next slide. So this is we call as N glucoside. Okay, just now in the previous slide we call as O glucoside, okay, with the glucose. But for the N glucoside is the conjugate with the amino acid. So amino acid, they have the nitrogen atom. So when they have conjugation with the amino acid, so become N-glucoside. So they cannot be hydrolyzed and serve as permanent inactive cytokinin. It's different to the conjugation with the glucose, which is they able to hydrolyze with the enzyme and it able to restoring back their active form. However, the difference in the amino acid, when they are conjugate with the amino acid, they are permanently inactive, that cytokinin. Similar also if the cytokinin conjugate with the alanine, they also produce a permanent inactive cytokinin. So if you look on A and B, they are towards to the N-glucoside, which is conjugate with the amino acid. However, for the C9-alanyl zetin is a product from the conjugation with the alanine. So all A, B, C is the permanently inactivating cytokinin, different with the previous slide regarding to the conjugation with the glucose. Okay, second mechanism that able to control the activity of the cytokinin we call as degradation. So degradation by the cytokinin oxidase or dehydrogenase, or we can call only as a CKX. So this CKX actually the name of the enzyme. So this enzyme is the degradative enzyme. So where, for example, if you look on the figure, isopentyladenine is one or example of the cytokinin hormone. So this isopentyladenine will be degraded, okay, by the CKX. become the 3-methyl-2-butanol and adenine. So it might be from the active, so become the inactive form because they already degrade, become from one molecule to two molecules. However, the uniqueness of this pathway, they only can be degraded for the cytokinin with the double bond in the isoprenoid site. Okay, so if you look on the isopentenyl adenine, they have the double bond. So because of that, they are able to degrade by the CK hex. Alright, now we go to the site of the cytokinin synthesis, which is in the simple words that we can say that this cytokinin they produce in the root tips, in the immature seed, and also in the developing fruits. So this is actually from the previous study. They stated these three locations synthesis more cytokinin. However, they able to transport to the another parts of the plant. Okay, now we go to the biological effect of the cytokinin. Biological effect of the cytokinin is actually about the role or about the function of the cytokinin. So the first role of the cytokinin is they involve in the cell division. So if you look on the illustration, that is about the cell cycle. You need to know that in order you to learn about the cell, you need to know about the basic information of the cell. Where the cell they have the event that we call as cell cycle. Where in the cell cycle they have two major processes, which is interphase and mitosis. So this cytokinin is involved in this cycle, which is we call this cytokinin as cell division factor that require for the cell proliferate. And this cytokinin only stimulate the cell division in the presence of auxin. Means they dependent also to the auxin hormone. And the specific role of the cytokinin is to regulate the progression of the cell cycle. So if, for example, the cytokinin is absent or the auxin is absent, so the cell culture will be only remain in the G1 or G2. Okay, so they not have the transition to the next phase. Okay, first you need to recall that in the cell cycle you have growth 1, G1, G2. Okay, S means synthesis phase. Okay, and M means the mitosis process. So the first process in the cell cycle is G1. So if the cytokinin or auxin is absent, so they only remain in G1 or only in G2, okay, without have the progression in the cell cycle. So in the presence of the cytokinin, they needed to active the cyclic dependent kinase. So this is the name of the enzyme, or we can call as CDK. So the activation of the CDK by the cytokinin will be allow cell to have transition from the G2 phase to the mitosis. Because in the cell cycle, cell will be undergo first the interphase process, and after that when all the ingredients have been enough, so they will enter the mitosis process. Okay, now we go to the example of this function. So for the previous study, they have been stated that in the role of the cytokinin, in order to regulate the cell division is happen in the Arabidopsis plant. So in the Arabidopsis plant, this cytokinin, they regulate the level of the T-type cyclin. So CYCD. Okay, as I mentioned before, this CYCD require also the auxin. Okay, they present of the auxin. So this CYCD type control the transition, okay, from the G1 phase to the S phase of the interface. So CDK is the enzyme that required by the cell to enter the S phase from the G1. Okay, second function is the cytokinin, they have the interaction with the auxin in order to regulate the formation of the root and shoot in the cell culture. So means when you look on the image, you can see that for this function of the cytokinin, you only can observe in the plant tissue culture. So you need to grow the explant and onto the synthetic media that you already mix with the two different hormone, which is cytokinin and also the auxin. So when you grow the plant, okay, onto the MS media, okay, the media for the plant tissue culture, if the ratio of the auxin to the cytokinin is too high, so the plant will be produce only the roots. However, if the ratio of the cytokinin to the auxin is too high, so that plant not produce the root but they produce the shoot. If you put in the media a proper ratio, means a similar concentration of the auxin and cytokinin, so they not produce root, they not produce also the shoot, but they regenerate, become a callus. So if you look on these four treatment in the cell culture, you can see that they have some explant produce more root, we call rooty, or they produce as a callus, or they produce a lot number of the shoot, we call as shooty. So we can see that the different ratio of the cytokinin and the auxin, which is we look first for the shooty. BA or BAP is the cytokinin 1, okay, and AA is 0, so means the concentration of cytokinin is high, okay, compared to the auxin. So because of that, they produce a lot, or they produce what we call as all the explant will be produced a shoot. However, if we look on the rooty explant, so they only put PAP 0 and NAA 0.05. So means do not have the cytokinin but have only auxin. So they not produce a shoot but they produce a lot of the root. So if you put a similar ratio or not actually far concentration on BAP1 and AA 0.5, so they produce a callus, and we compare with the explant that you grow onto the control media, which is means do not put any plant hormone, is only grow by using the micronutrient and macronutrient that you formulate in your MS medium. Okay, this one is the image, okay, for the callus, and the callus that produce a shoot. So means in the callus treatment, they only have the similar ratio. However, the next culture that have the shoot means they also produce or they put some concentration of the cytokinin. Okay, for example, BAP. Alright, so now we look on this function or this role of the cytokinin. They serve as the antagonize. So means they antagonize to the effect of the auxin, okay, on the apical dominant. Okay, so these phenomena Okay, it's because of the parasite, which is a fungus, okay, that cause the we call as phenomena as a witch broom. So this witch broom is when that tree they produce over, or they stimulate overproduction of the cytokinin. They produce a lot of the cytokinin. So because of that, they're releasing the axillary bud from their dormancy. So in order to reduce this witch broom to occur, they will inject the exogenous auxin in order to overcome this apical dominant. Okay, which broom we call also as the apical dominant because they have a lot of the leaf, okay, at the apical parts. Alright. So this witch broom, as I told before, that is because of the fungus. So the fungus, the name is Taprina betulina. The next function of the cytokinin is they involve in the formation of crown gall tumor. So some of the plant you can see from your outside of your house, you can see they may have the tumor. They may have the swelling form at their branches or at their stem. Where these swelling we call as crown gall tumor. So this gall tumor is because of the bacteria Agrobacterium tumefacium. And these bacteria actually they carry, why they able to produce a tumor, because in the gene of this bacteria they carry the tumor inducing plasmid, okay, that really responsible for the inducing the neoplastic growth of the gall tumor. So when they are infect the plant, it will be enter the genome of the plant and induce the plant to produce the tumor. Here this one is the example, maybe you can see before. So for the bacteria that have been transmitted, okay, their plasmid to the DNA of the plant, okay, you need to know some terms, which is transfer DNA, we call as tDNA, okay, from the plasmid of the bacteria. It will be incorporate with the host plant genome. And this tDNA, they contain 3 genes. Okay, for your information, why in this part you learn about the function of the cytokinin in the gall tumor production. Because in the tDNA, they contain 3 genes, which is 2 genes are necessary for the synthesis of the auxin and cytokinin. Okay, and the third gene, they are helpful to produce a nutrients for the bacteria we call as opines. So these kinds of regulation, okay, cytokinin, okay, with the A tumefacium will be actually reprogram the plant, okay, if they are infected, okay, by the bacteria A tumefacium. Alright, now we look on the function of the cytokinin as the inhibitor for the senescence. So in this function, how the cytokinin is the inhibitor for the senescence is we may look on the experiments that expression of the agrobacterium gene for the synthesis of the cytokinin, where in this experiment they put the leaf that have been infected by the agrobacterium uh, in the treatment that have the heat shock promoter, which is the treatment where they apply the heat in order to produce or increase. They produce and increase the zetin concentration. And actually, from the final result, when they produce high concentration of zetin, okay, from this treatment, they are able to delay the senescence process. Senescence mean is if you look of your plant, okay, when your plant have the increasing of their aging, and they will falling all the leaf, or the leaf become the color like a brown to black. So they able to delay that process. Okay, if they have high concentration of the cytokinin hormone. Now we look on this role for the cytokinin. Cytokinin also will be stimulate or influence the distribution of the nutrient in the tissue. So if we apply the cytokinin to the leaf, if you look on this figure, so that nutrient will be migrate, okay, migrate to the spot where the application was made. Right? So how actually or why this migration of the nutrient occur is believed that this migration occur because cytokinin will be stimulate the metabolism. Okay, and because of that the local cell become as a sink for the nutrient. Sinks means the source of the nutrient, okay, for another cell of plant. Okay, this one is the example of the distribution of nutrient. Okay, now we look on the function of the cytokinin in maintaining the shoot meristem. So this plant hormone is also maintaining the shoot meristem, where shoot meristem is the part in the plant where they have high shoot production. So if cytokinin is low or reducing in vivo cytokinin concentration, in vivo mean endogenous cytokinin, so it may be cause that plant retard their shoot development, dwarf or reduce size of the shoot meristem, or maybe they have a slow formation of leaf primordia. So you need to know that leaf primordia actually the leaf, a new leaf at the apical parts, and they may also reduce the number of the leaf. So in this function, you will look on the name of the gene that encode for the function of this cytokinin in order to maintaining the shoot meristem. We call as lonely guide gene or log gene. So this log gene encode for the phosphoribohydrolase that regulate the meristem activity, okay, by converting inactive cytokinin to active phase. Base form means from the inactive cytokinin, so it will become the active cytokinin in order to regulate the shoot meristem. So because of that, they also involved in the maintaining the shoot meristem for the cytokinin. Alright, now we look on the second gene, okay, involved in the maintaining the shoot meristem, okay, under the cytokinin, which is second gene we call as KNOX gene. Okay, they regulate the synthesis of the gibberellin and cytokinin. So for these genes, they have the heme we call as homobox or homodomain sequence. And these homobox genes is have the fundamental roles in controlling the cell developments. And they also have the protein we call as KNOX protein. if they have some mutation happen in the KNOX genes, so it may affect the development of the meristem. So what happened? Um, if we look on this function, they are for the cytokinin to maintaining the stem, shoot meristem. So if we look on here, this gene actually expressed for the cell of the shoot apical meristem, but not in the leaf primordia or developing leaf. So if, for example, the function of the KNOX genes have been mutated, so it will be expressed in the developing leaf, where they produce the knock of the cell, or we call as ectopic shoot form. Okay, so this ectopic shoot form is actually they not produce a normal leaf, but they produce only a small leaf. Okay, a small leaf and like a blade. Because why? Effect from the mutation of the KNOX gene, it will be result the accumulation of the KNOX protein. When this accumulation of the protein, they may cause the cell continue dividing, but rather undergoing the differentiation. Means they continue divide, but they do not differentiate become the normal leaf. Okay, that's all for today.